Hello, my name is Maria, and today I'm going to present to you Miconia Calvescens biological control actions and strategies. A quick overview, I will be recapping the biological control of Miconia calvescens on the Hawaii Island, followed up by a vision statement of protecting and restoring native lands. Next, going into the adaptive management plan, starting with the assessment stage, following up with the success criteria and evaluation stage, finishing up with the management alternatives in the adjustment stage. So quick recap of the biological control of Miconia calvescens on the Hawaiian Islands. So the Miconia species was introduced to Hawaiian Islands as a botanical plant in the early 1960s. It is a fast-growing tree that's capable of producing millions of seeds annually and is widespread by humans, birds, and animals. Miconia trees grow in close proximity, thus suffocating other plants around it. It has been identified as an invasive species and is threatening well-being of approximately 70 other plant species in the Hawaii Islands alone. In order to prevent further damage and reverse some of the effects, biological control is necessary. For this purpose, Euthanasia crassipi is a proposed natural predator for biological control of the Miconia. Vision statement still remains protecting and restoring native lands. On the right side, you can see a graphic of the adaptive management plan I'll be following, starting with assessment, going into implementation, followed by monitoring, evaluation, adjusting, and then implementing and assessing new ways forward. So currently, we are at the assessment stage. We are lab testing crassipi effects on the Miconia calvescens. Following that, we'll go into the implementation stage of shipping the crassipi larva from Costa Rica and releasing natural predator into affected areas of the island. After that, we'll monitor the effects of natural predator on the pest species. Next, we will evaluate if the natural predator is effective and is it reducing or inhibiting spread of the pest species. Finally, we'll go into adjustments. After evaluation, we'll take adjustments as necessary if the desired species effects are not met. So during the assessment stage, on the right side, you see a graphic which has biological and rearing identifications of the Miconia calvescens, proposed biological control, some of the host specificity, and other graphics. So the crassipi is a gregariously feeding larva. In lab testing, approximately 60 to 80 larvae will consume several hundred square centimeters of leaf tissue, which is approximately to one leaf of the Miconia. The damage generally distributed across several leaves as the species likes to move around. Study among 73 species showed that crassipi larva prefers feeding on only and will survive only on Miconia calvescens plant. Finally, of note, in natural habitat, crassipi does not have extensive defoliation on the species, and that's presumed due to natural enemies present in the environment. So, success criteria during the evaluation stage, the release areas will be reevaluated and monitored biannual, and the success will be measured by percent of Miconia calvescens coverage eradicated and reduction of pest plants spread to new areas. Following the evaluation and success stage, we'll go into adjustments. Depending on the outcomes of the evaluation, there might be an alternative biological control measures needed that will go back into the assessment stage where we'll be testing and assessing success of alternative natural predator. Or if other means are necessary, there'll be a chemical method tested as well. Previously, the triclopyr is used against invasive plants and is spread over the area by plan. It has previously been used for Miconia calvescens mitigation on Hawaii Islands, and despite some success using this chemical control specifically, the species continues to proliferate. Another alternatives can be mechanical methods. This would, however, be heavily relied on volunteers and DLNR involvement. And finally, it's not feasible for current proliferation of the Miconia calvescens and how widely it is spread across the islands. And the last one will be no alternatives. These are my references. Thank you.